Hey guys, welcome back to a new biology with Zangshan video. So today we are going to do the Cambridge IGCSE Biology Paper 4 Theory Extended, the October-November 2023 series, Paper 4 Variant 3. So this paper is considered to be the hardest paper in this October-November series, which is shown by its low threshold. So for those who watch the paper 4 variant 2 video, right, there are a few mistakes that I have made. Make sure you check it out, okay, so that you don't get confused, and I will try my best to reduce as much mistakes as possible. So let's start off with this video, shall we? Okay, question 1a. Figure 1.1 is a diagram of an insect pollinated flower. So they give you the parts labeled A, B, C, etc. And part 1 asks you to use the information in figure 1.1 and complete the table itself. For structure A, it's pointed here, which is showing the stigma. Okay, so what is the function of the stigma? Is just to trap the pollen during pollination, basically. Okay, for B, this is pointing the petals, so just use a very simple function that it helps to attract insects. For F, they want this part, which is actually the sepal. The sepal helps to protect the buds of the flower. Okay, part 2. State the names of the two structures that form the stamen in a flowering plant. So stamen is basically the male reproductive system. So what are the two things, which is anther and the filament? Okay. Alright, B part 1. Describe the stages in the reproduction of a flowering plant from self-pollination to fertilization. So this is 5 marks here. So how am I going to, write this response is by going step by step. I will first answer self-pollination, then to fertilization. So for self-pollination, just very simple, is the process where it happens on the same plant of the same species. I will get a 1 mark from here. The next one is I describe the process of fertilization, so which is the pollen transferred from the anther, then to the stigma of the same plant, which will get me the first mark, which allows the pollen tube to grow. Another mark is formed here. The pollen tube then travels down the style and moves into the micropyle. Another mark is formed here. The pollen tube nucleus fuses with the nucleus of the ovule, where a zygote is formed. Another 1 mark. So very simple. I feel is that you do not need to write a lot for self-pollination. Write as detailed as possible for fertilization, because that's where all the majority of the marks is from. Okay. Part 2. Outline the advantages and disadvantages of self-pollination compared with cross-pollination. So this is 4 marks and asked for advantages and the disadvantages. So I give 2 points from here and 2 points for disadvantages. So for advantages, I have chosen higher chance of successful pollination and less pollen wasted. Okay, and the disadvantages is that this would decrease the genetic variation, and of course a much higher chance and a susceptible risk of gaining genetically inherited disease. Okay, so this is how I get the full 4 marks. Try to remember some advantages and disadvantages for both pollination so that you are able to write it down. Okay, question 2a. Figure 2.1 is a diagram of a cross-section of a part of a leaf. So they give you this, basically is what is inside a leaf, basically. So identify and explain how the structure labeled J and K are adapted for photosynthesis. So they give you 4 marks. So the first thing in this kind of question is to identify what is J, identify what is K. So for J, I have is the upper epidermis, which gives me 1 mark. Alright, and then your explanation is that it will be allowing light to enter through the leaf so that the palisade mesophyll cell is able to undergo photosynthesis. For K, it will be the xylem vessel, okay, which basically transport water and mineral ions. Okay, just a very simple structure that helps with photosynthesis. Okay, B. In an experiment, the tail crest plants or Arabidopsis Thalliana were grown in a normal atmospheric and high carbon dioxide concentration. The transpiration rate, the mean number of chloroplasts per cell, and the concentration of starch and magnesium ions were measured. The results are shown in table 2.1. So just have a look at this, and they are asking you to complete the sentence, almost like fill in the blanks. So table 2.1 shows that increasing the carbon dioxide concentration causes more starch to be produced in the leaves. This shows that at a normal carbon dioxide concentration, carbon dioxide will be a limiting factor. So we have a keyword that it is the carbon dioxide concentration which caused the change. Therefore, it will become a limiting factor. During photosynthesis, 6 molecules of carbon dioxide are required to make 1 glucose of molecule. So this is corresponding to your understanding of the photosynthesis equation, which is 6CO2 plus 6H2O, which will form C6H12O6 plus 6O2. So 6 molecules of carbon dioxide will form 1 molecule of glucose. Okay, the greater quantity of starch stored in the leaves grown in a high carbon dioxide concentration means when needed, more sucrose can be produced for transport in the phloem. So the leaves act as a source. So why is it a source? Because it will be used to transport. So the source will help to source it out for the plant's needs. The greater number of chloroplasts per cell in the leaves grown in the higher carbon dioxide concentration means that more energy can be absorbed from light and transferred to chemical energy. The transpiration rate is lower than the carbon dioxide concentration is higher. This means that reduced loss of water vapor from the leaves. So when you stuck on what am I supposed to put, just see from this one. When the moment there's a mention of word a transpiration, you must have the word of water vapor happening, because the plants don't leave the, like the leaf cell itself, it doesn't leave in the form of liquid water, but instead it's in the form of water vapor, which is basically gaseous water technically. Okay. And then lastly, magnesium ions concentration is lower in these leaves because they have used the magnesium ions to make chlorophyll. This is testing on your mineral requirements, okay? So that is all for 7 marks. Question 3a, part 1, complete table 3.1 by writing in the percentages of carbon dioxide and oxygen in inspired air and in expired air. For this question, I am requesting you to memorize these values. So you must know that carbon dioxide is up now and oxygen is second, so there will be 0.04% inspired air and 21% of oxygen, 4% in expired air and 16% in oxygen. Okay, so question 2, a scientist measured the number of dust particles in inspired air and in expired air. They found fewer dust particles in expired air. Suggest a reason for their observation. So this is coming back indirectly to how does the mucus play a role in the throat or in any system that in the alimentary canal. So for this case, they say that there's lesser particles when it's being exhaled. So this means that all of the dust particles has already been trapped by the mucus itself. Okay. Part B. Figure 3.1 is a diagram of alveoli and associated blood vessels. So they give you, this is actually located in the lungs. So part 1 says explain how the structure of capillary is related to its function. So when they say structure of a capillary, please don't tell me what does a capillary do. They want to tell you that what makes this capillary so efficient, and it's because of its structure. So the structure of a capillary is that it's one cell thick. Always capillaries is one cell thick, which helps to allow the efficiency of transport of valuable substances like oxygen and nutrients to tissue and the rest of the body. And it also has a very small lumen, providing a shorter diffusion distance. So since this is 3 marks, right, always have one cell thick, small lumen and a short diffusion distance. Then you get your 3 marks really, and just elaborate a little bit why does this helps. Okay. Part 2. State the name of the component of blood that transport oxygen. So the component of blood, which is actually all the 4 things that's inside, with the platelets, the plasma, the red blood cell, the white blood cell. So for this case will be the red blood cell. Part 3. State the name of the blood vessel that transport blood from the heart towards the capillary in the lungs, which is the pulmonary artery. Part 4. State the location and the function of the cartilage in the breathing system. So there are multiple answers I have given you, which you can put the trachea, the bronchus, or the larynx. It helps to prevent the collapse of the airway, or as simple as protect the vocal cords. Okay? Alright, C. A student measured the rate and depth of breathing of an athlete for 30 seconds at rest. Okay, so the data shown in figure 3.2 shows the volume of air breathed in and out in dm³ against time and seconds. So part 1 says using the information in figure 3.2, calculate the rate at breathing at rest. So it's told you that one breath is between this to this. So calculate the rate of breathing rest per minute. So for this is 30 seconds, you count as 1, 2, 3, 4, 5, 6, and about 6.5. So since this 30 seconds is 6.5, 60 minutes should be times by 2. So the answer is 13 breaths per minute. The measurement were repeated while the athlete was running on the treadmill. The data are shown in figure 3.3. Using the information in figure 3.3, calculate the volume of air inspired in one breath from 25 seconds. So for this question, it may seem very confusing at first, but it's actually at 25 seconds. Alright, I will be using this value minusing the lowest value, which I will get 2.6 in the end. Okay? Part 3. Explain the effect of exercise on the rate and depth of breathing shown in figure 3.2 and figure 3.3. So what happens when you exercise? What's going to happen to the rate of breathing that's going to happen? So it will definitely increase. Why? Because the body requires more oxygen to continue aerobic respiration so that enough ATP is released for muscle contraction. It is also helpful for the quick and efficient removal of carbon dioxide out from the body. Okay? So this is very simple, 4 marks, Okay, question 4a. Complete the sentences about the control of body temperature. So the human body maintains a constant internal temperature. This is the key word here already. This is an example of homeostasis because it's technically the definition is about here. Okay, and when the temperature moves away from the set point, the mechanism of a negative feedback returns the temperature to the set point. So what does the negative feedback mean? If there's an increase in temperature in my body undergoes negative feedback and tries its hardest to make sure the temperature is back to its normal state. Otherwise, there will be a lot of complication in the body, for example. Okay, so part B, figure 4.1 is a diagram of a section of a human skin. Similarly to the first question, they've given you the parts and label it, and then they want you to name it. So for L, this is referring to here, this is the sweat glands. Receptors is O, okay, you can see it looks like some sort of spike, this is a receptor. And for J, which is referring here, is actually the hair erector muscle. We can also accept erector muscle itself. Okay, alright, part 2, describe how humans maintain a constant body temperature when the external temperature decreases. So don't mention negative, it's kind of basically negative feedback, but then they want to tell you like how does the steps to maintain the temperature again, and how does it detect. So firstly, the receptors will definitely detect the decrease in the external temperature, where the nerve impulses will be sent to the brain quickly and producing a response. So when a response is generated, the hair on the skin will definitely stand up, which is caused by the hair erector muscle, which helps to insulate by trapping air. The arterioles in the skin will also begin to vasodilate, which helps to stop the production of sweat from the sweat glands. Okay, so this is 6 marks, quite a lot, but then just use your understanding what happens when you're feeling cold, such as during the exam, your hair begins to stand up. So all of these is relatable. Okay, C. Blood glucose concentration is maintained at a constant set point using the hormones glucagon and insulin. So where is this coming from? It's from the pancreas. And describe the effect of glucagon on the body is that it definitely helps to break down the glycogen in our body into glucose, and this will help to increase the blood glucose concentration in the bloodstream. Okay, question 5a, state two cell structures found in both animal and bacterial cell. So I've just chosen two random, one with the DNA and the cytoplasm. We can also definitely accept the cell membrane and the ribosomes. These are all accepted too. B, state why bacteria are useful in biotechnology. So these are just a few that I chosen just to achieve the mark, which is rapid reproduction rate, no ethical concerns, and presence of plasmids. Okay, so this is the 3. And C, genetically modified bacteria were grown in a fermenter. The number of bacteria was measured and the data are shown in figure 5.1. So this is technically a population growth curve. So on part 1, they say to draw an X to identify the lag phase. The lag phase is actually before the log phase, which is actually here. Okay, and next one, I say draw a Y to show where the birth rate is equal to the death rate, which actually is the stationary, which is actually located here. And part 3, they say calculate how long it takes for the number of bacteria to reduce by half after the bacteria have been in the fermenter for 24 hours. So it will take 12 hours definitely, and the whole thing starts to half itself. Okay. And now part 4, they say describe and explain the change in bacterial population size from 24 hours to 50 hours shown in figure 5.1. Of course, when you see after 24 hours to 50 hours, right, it has entered the death phase. So this will definitely decrease the bacterial population size. So there could be multiple reasons where how this happened is from the increased competition, and there's probably a lack of nutrients required, and also the ability to survive has already decreased due to all these kind of factors. Okay, and V is the fermenter is kept at the optimum temperature for the bacteria. Why is this important for enzyme function? So that when increased temperature, it helps to increase the kinetic energy. So this helps to ensure that there's enough kinetic energy for the formation of maximum enzyme substrate complexes being generated, which helps to ensure the high rate of reaction is still maintained. For part 6, they say state why the bacteria are grown in a liquid that contains amino acids. We know that amino acids are the building blocks of protein. So when amino acids are present, the bacteria will be using it to make proteins, which can definitely significantly help for growth. Okay, and question 6a, they say xerophytes are plants that are adapted for an environment which has very little available water. So describe the meaning of adaptation is it's always a result of natural selection. So because if natural selection doesn't happen, the organisms are unable to adapt to the surrounding. So whereas population will now begin to be better suited to the environment for over many generations. Okay, and B, figure 6.1 is a photograph of a saguaro cactus or Carnegie gigantea, which lives in the desert. The climate in the desert has a very low rainfall and very high daytime temperatures. So describe two visible adaptive features shown in figure 6.1 and explain how each feature of it benefits in the desert. So the feature 1 of a cactus obviously will be the spikes or the thorns itself, which helps to prevent the water loss, and definitely it protects against consumers like the herbivores or the carnivores. Actually, typically it's the herbivores and the omnivores usually. And feature number 2 is definitely its thick stem. You can see the thick stem is located here, which is very important for the storage of water. Part C, table 6.1 shows some data about stomatal density in the leaves of one plant that is not a xerophyte and 3 xerophyte plant. So using the information in table 6.1, estimate the total number of stomata in an ice plant. So it's located here with a lower leaf surface area of 8 centimeters square. The keyword is to tell you how many stomata are there. So we know that it's 42 on the lower leaf surface. But however, the surface area given is in centimeters square. Therefore, I have to convert this into millimeters square, which is by times 10 and times 10 again, which is 800 millimeters square. Then I use the 800 millimeters square to times by 42, and then I will get 33,600 stomata. Part 2, explain the data shown in table 6.1. So for overall from the data I've seen is that the lower leaf surface have more shelter from the wind. Okay, therefore this helps to significantly reduce the rate of the transpiration rate. And again, there is a low presence of stomata from the 3 xerophytes. Why? Because they are all trying to prevent the loss of water. And lastly, there are xerophytic forests which are threatened by human overexploitation. So just reason why it's important to conserve xerophytic and ecosystem, just give a very logical answer, is to maintain the biodiversity and also to ensure the stability of the food chain remains, because cactus is a producer. So without producers, then there's an extinction happening. This will definitely significantly affect the food chain. Alright, so thank you so much for listening, guys. See you guys next time. Bye bye.